Welcome back to the Deep Dive. Today we're taking a deep dive into the intricate world of the brain, using Dale Purves' Neuroscience, 3rd edition, as our guide.
It's like a cheat sheet to understanding how your brain really works, breaking down those complex concepts.
Exactly. We know neuroscience can be, well, a little intimidating, but trust us, stick with us, and you'll be amazed by what you discover.
Absolutely.
So let's start at the very beginning. How did scientists even figure out what the basic building blocks of the brain are? What it's all made of?
Well, early researchers faced a huge challenge because, unlike many other cells, neurons have these crazy complex shapes.
Oh, I can imagine.
Yeah, with branches going everywhere, it was incredibly difficult to figure out if they were individual cells or all part of a single interconnected network.
So like a tangled mess of wires, but on a microscopic scale.
Exactly.
So how did they finally solve this puzzle? Was it a new technology or a brilliant insight?
It was a bit of both, actually. Camillo Golgi developed this amazing staining technique that allowed researchers to visualize neurons under a microscope.
Oh, that's got to be a game changer.
Yeah, it was huge. But Golgi himself believed in something called the reticular theory, the idea that the nervous system was one giant web of interconnected cells.
So he thought it was all one continuous network.
Right. But then came Santiago Ramon y Cajal and Charles Sherrington, who championed the neuron doctrine.
Which basically said the opposite.
Exactly. They argued that the nervous system was made up of distinct individual neurons, not a continuous network.
Okay, so two competing theories. How did they finally settle the debate?
It took even better imaging techniques to finally prove that the neuron doctrine was correct.
It's amazing how those early pioneers figure this out with limited tools.
It really is.
Now, the book talks about how diverse neuronal structures are. Can you tell us more about that? Why is it significant?
Well, this diversity really highlights the incredible adaptability of neurons. Some neurons, believe it or not, don't even have dendrites, those branch-like extensions that receive signals.
Really? No dendrites at all?
Nope. While others have dendrites so complex, they look like a mature tree with branches reaching out in every direction.
Well, that's a pretty wild visual.
It is.
I'm guessing that structure plays a big role in how neurons function.
Absolutely. A neuron with a simpler structure, one without many dendrites, might only receive signals from a handful of other neurons.
Okay, so limited input.
Right. But a neuron with that intricate tree-like structure could be receiving input from hundreds, even thousands of other neurons.
So it's like a communication hub, handling massive amounts of information.
Precisely. And that's what allows the brain to process information in such a nuanced and sophisticated way.
It's mind-boggling to think about all those signals flying around.
It is.
This makes me wonder, how do we actually observe this intricate dance of neurons in action? How do we peek inside a working brain?
Well, that's where fMRI comes in. It's completely revolutionized how we understand the brain.
fMRI. That stands for functional magnetic resonance imaging, right?
Yep, that's it. Unlike older methods like PE scans, fMRI doesn't use any radioactivity.
So it's safer for repeated observations.
Exactly. That means we can observe brain activity in real time, over and over again, in the same individual.
We can literally see the brain lighting up as someone thinks or performs a task.
You got it.
That's amazing. What kind of detail are we talking about? Can we see individual neurons firing?
Not quite individual neurons, but we can pinpoint activity down to a few millimeters and track changes over seconds.
So we can see which areas of the brain are engaged during different activities.
Exactly. For example, we can see how the brain engages differently when someone is solving a math problem versus, say, listening to music or experiencing a strong emotion.
It's like having a window into the working brain.
Precisely.
Before we get too deep into the complexities of the human brain, I was fascinated by the book's detour into the world of squid. It talks about their giant nerve cells. Why are those so important for neuroscience?
Yes. Those fascinating creatures with their giant axons. These cells are hundreds, even thousands of times larger than the axons in mammals.
Wow, that's huge. Why is this size difference so important?
Well, their massive size made them the perfect subjects for early neuroscience experiments.
Okay, I see. Bigger is better in this case.
Exactly. Researchers could actually insert electrodes directly into these giant axons to measure electrical activity.
Really? Directly into the axon?
Yeah. It's like having a much larger, more accessible electrical circuit to study.
That's incredible. So they were kind of like the guinea pigs of early neuroscience.
In a way, yes. And it led to some major breakthroughs, including the very first recordings of action potentials.
Now, action potentials. Those are the fundamental electrical signals that neurons use to communicate, right?
Precisely. And understanding them is key to understanding how the brain processes information.
They're like the language of the nervous system.
Exactly.
So let's break it down. What exactly happens during an action potential? What triggers it?
Well, at rest, a neuron's membrane is more permeable to potassium ions, giving the inside of the neuron a slightly negative charge compared to the outside.
So it's like a tiny battery storing electrical potential.
Perfect analogy. Now, when a neuron receives a signal, it causes a small depolarization.
Meaning the inside becomes a bit less negative.
Exactly. If this depolarization hits a certain threshold, it triggers a rapid influx of sodium ions, making the inside of the neuron suddenly positive.
So it's like flipping a switch, reversing the electrical charge.
Precisely. That sudden change in electrical potential is the action potential,
And that signal races down the axon.
like a chain reaction, carrying the signal to other neurons.
Wow, that's incredible. But this influx of sodium can't last forever, can it?
You're right, the membrane quickly becomes less permeable to sodium and more permeable to potassium again.
Okay, so it goes back to its resting state.
Exactly. Potassium ions then flow out, restoring the negative charge inside the neuron, ready for the next action potential.
It's a remarkably elegant system. But what controls these gates that regulate the flow of ions in and out of the neuron?
Those gates are actually ion channels, specialized proteins embedded in the membrane.
They're like tiny doorways for specific ion.
Precisely. Some channels are specific to sodium, others to potassium, calcium, or chloride.
So it's a very controlled process, not just a random flow of ions.
Very controlled. The opening and closing of these channels are influenced by changes in membrane potential, the binding of neurotransmitters, even mechanical forces.
Wow, so many factors at play.
Many factors, all working together. This precise control of ion flow is what allows neurons to generate and transmit electrical signals, forming the basis of everything the brain does.
It's amazing how such tiny channels can be responsible for so much. But now let's zoom out a bit and look at how neurons communicate with each other.
Okay?
Because action potentials are just one part of the story, right?
Yeah.
The real magic happens at the synapse, the junction between two neurons.
Exactly.
Can you tell us more about what makes the synapse so special?
Well, the synapse is where the electrical signal of the action potential is converted into a chemical signal.
So it's like a translator, converting messages between different languages.
In a way, yes. When an action potential reaches the end of an axon, it triggers the release of neurotransmitters from tiny sacs called synaptic vesicles.
Neurotransmitters, those are the brain's chemical messengers, right?
Right. They travel across a tiny gap called the synaptic cleft and bind to receptors on the receiving neuron.
So it's like a tiny postal service, delivering messages from one neuron to the next.
Precisely. And this binding can either excite or inhibit the receiving neuron, influencing whether or not it fires its own action potential.
So it's a chain reaction, one neuron signal influencing the activity of the next.
Exactly.
It's remarkable how this communication happens so quickly and efficiently.
It is. Now, you might be wondering, how do we even know that neurotransmitters are released in discrete packets?
It seems impossible to study something so tiny.
That's where studying the neuromuscular junction was really helpful. Katz and his colleagues noticed these tiny, spontaneous depolarizations called miniature end plate potentials, or MEPP.
Mini potential.
Yeah, mini, because they were much smaller than the full-blown end plate potentials that actually trigger muscle contractions.
Interesting. So what causes these mini signals?
Well, they found that both the miniature and the full-blown potentials were sensitive to drugs that blocked acetylcholine receptors.
Acetylcholine, that's the neurotransmitter at the neuromuscular junction, right?
Exactly. This finding suggested that acetylcholine wasn't released in a continuous stream, but rather in discrete packets.
Like little bursts of chemical information.
Exactly. Each MEPP represents the release of a single quantum of acetylcholine.
So it's like the difference between a single raindrop and a sudden downpour.
A great analogy. And when a neuron is stimulated, it releases many of these quanta simultaneously, resulting in a larger signal.
This brings up another question. How are these packets of neurotransmitters packaged and released? It seems like a very delicate operation.
It is. And that's where synaptic vesicles come in. These tiny membrane-bound sacs are filled with neurotransmitter molecules.
So they're like tiny containers ready to be shipped out.
Precisely. And they go through a carefully choreographed cycle to make sure neurotransmitters are released in a controlled and efficient manner.
Okay, so let's break down this synaptic vesicle cycle. What are the main steps involved?
Well, first, the vesicle has to dock at the presynaptic membrane.
Like a ship pulling into a port.
Exactly. This involves proteins called snare proteins, which act as tethers, pulling the vesicle close to the membrane.
So the vesicle is ready to unload its cargo of neurotransmitters.
Right. The next step is priming, where the vesicle gets ready to fuse with the membrane.
So it's prepped and ready to go.
Yes, and this involves other proteins, including one called synaptoctamin, which acts as a sensor for calcium ions.
Calcium, what does that have to do with this process?
Calcium is the key. When an action potential arrives at the axon terminal, it opens calcium channels and calcium ions flood in.
Okay.
This influx of calcium triggers the vesicle to fuse with the membrane and release its contents.
So calcium is like the signal that says go. And what happens to the vesicle after it releases its neurotransmitters? Does it just disappear?
Nope. The vesicle membrane is recycled back into the neuron through a process called endocytosis.
So it's like a reusable shopping bag?
Exactly. This ensures a constant supply of vesicles ready to be filled with neurotransmitters and sent out again.
That's amazingly efficient. But what happens to the neurotransmitters once they're released into that tiny gap, the synaptic cleft?
Well, they don't just float around aimlessly. They have a crucial job to do. They bind to receptors on the postsynaptic neuron, the receiving neuron on the other side of the synapse.
So the message is finally delivered. What happens when those neurotransmitters bind to their receptors?
That's where things get really interesting because there are different types of receptors.
Oh.
Yeah, the two main classes are ionotropic receptors and metabotropic receptors.
Okay, two different types of receptors. How do they differ?
Ionotropic receptors are like fast-acting switches. When a neurotransmitter binds, it opens a channel, allowing ions to flow into or out of the receiving neuron.
So it's a direct and immediate effect.
Very direct and very fast. That speed is what's responsible for things like muscle contractions and rapid sensory perception.
Wow, so it's all about speed with ionotropic receptors.
Exactly.
And what about metabotropic receptors? Are they slower?
They are. They don't directly control ion channels. Instead, they trigger a cascade of intracellular events.
So it's more of a chain reaction.
Yes. It can lead to changes in ion channel activity, gene expression, and a whole bunch of other cellular processes.
Okay, so these receptors are involved in more complex long-term effects.
Precisely. Think about things like learning, memory, mood regulation.
It makes sense that those processes would involve more intricate signaling pathways.
It does.
It's fascinating how the brain uses these two types of receptors to achieve such a diverse range of effects.
It's a remarkable system.
It really is. But now I'm curious about how the brain takes all this neural activity and makes sense of the world around us.
Ah, yes. We're entering the fascinating realm of sensory perception.
All that information from our senses, sight, sound, touch, taste, smell, it all gets transformed into meaningful experiences.
Exactly. And it's happening constantly. We're bombarded with sensory information every second of the day.
I can't even imagine how the brain keeps up with it all.
It's an incredible feat of processing. One key principle is that the type of sensory stimulus is determined by which specific receptors are activated.
So the brain knows whether it's dealing with light, sound, or touch based on which receptors are sending signals.
Precisely. And also where in the brain those signals are sent.
So it's like a complex map with different regions of the brain specialized for processing different types of sensory information.
Exactly. For example, our sense of touch relies on a variety of mechanoreceptors.
Those are the receptors that respond to mechanical stimuli like pressure, vibration, and stretch, right?
You got it.
And those mechanoreceptors are distributed all over our skin. But some areas are much more sensitive than others, like our fingertips, for example.
That's because your fingertips have a much higher density of mechanoreceptors compared to, say, your back or your legs.
So the more receptors, the more sensitive the area.
Precisely.
It's amazing how that difference in receptor density translates into such a dramatic difference in our perception of touch.
It is.
But how does the brain know which part of the body is being touched? Like, how does it know that it's your finger feeling the texture of something and not your elbow?
That's thanks to something called the somatosensory map in the brain.
Somatosensory map.
Yeah, imagine a map of your body, but distorted, where the size of each body part reflects its sensitivity to touch.
Okay, so it's not geographically accurate, but more like a sensory map.
Exactly. Your fingertips, lips, face, those highly sensitive areas, they have a much larger representation on the map than less sensitive areas.
It's fascinating how the brain prioritizes different parts of our body based on their sensory importance.
It is.
But touch is just one of our senses. What about pain? How does the brain process pain signals?
Pain is a fascinating and crucial sense because it alerts us to potential tissue damage and helps us avoid further harm.
It's our body's way of saying, hey, something's wrong.
Exactly. And pain perception relies on nociceptors, specialized receptors that detect potentially harmful stimuli.
So nociceptors are like the body's alarm system.
That's a great way to think about it. And there are different types of nociceptors that respond to different types of stimuli, like mechanical pressure, extreme temperatures, and chemical irritants.
So there's a nociceptor for every type of pain-inducing experience. But how does the brain know whether it's a sharp, immediate pain like a cut versus a dull, throbbing pain?
That has to do with how the pain signals travel from the nociceptors to the brain.
Oh, tell me more.
There are two main types of nerve fibers involved, A delta fibers and C fibers. A delta fibers are fast-conducting and myelinated.
Myelinated, meaning they have that fatty insulation that speeds up signal transmission.
Exactly. They're responsible for that sharp initial pain you feel, like when you stub your toe.
Ah, that makes sense. So what about C fibers?
C fibers are slower and unmyelinated, and they carry those lingering, duller pain signals.
So it's like a two-pronged alarm system. The A delta fibers send the initial alert, and then the C fibers provide a more sustained warning signal.
Precisely.
It's incredible how much complexity goes into something we often take for granted, like feeling pain.
It really is.
Let's shift gears and talk about vision. How does the brain create visual experiences from light?
Vision is an incredibly complex process that starts with the eye, which acts like a camera, capturing light and focusing it onto the retina.
And the retina is that light-sensitive layer at the back of the eye, right?
Yes. It's where the magic of converting light into neural signals happens.
And that conversion is done by photoreceptors, right?
You got it. Photoreceptors are specialized cells in the retina that contain light-sensitive pigments. We have two types: rods, which are responsible for dim light vision, and cones, which are responsible for color vision and fine details.
So rods are like our night vision goggles, while cones allow us to see the world in high definition and in full color.
A perfect analogy. And when light hits these photoreceptors, it sets off a chain reaction that ultimately leads to changes in the electrical potential of the cell, creating a neural signal.
So it's like light is transformed into electricity, which is then sent to the brain.
Exactly.
Does that signal go directly from the photoreceptor to the brain?
Not quite. The retina itself does some preliminary processing, refining and organizing the visual information before sending it off to the brain.
So the retina is like a mini brain, pre-processing the visual data before passing it on to the big boss.
Exactly.
Okay, so then what happens? How does that processed information get from the retina to the brain?
That's the job of the optic nerve, a bundle of nerve fibers that carries those signals.
Ah, the optic nerve. I remember learning about the optic chiasm. That's where the optic nerves from each eye partially cross over, right?
You remember correctly. The optic chiasm is crucial for ensuring that information from the left visual field goes to the right side of the brain, and vice versa.
It's like a traffic intersection, directing the flow of visual information to the correct destination.
Precisely. From the optic chiasm, the signals travel to a relay station in the thalamus called the lateral geniculate nucleus, or LGN.
The thalamus, that's like a central hub for sensory information in the brain, right?
Exactly. And from the LGN, the signals are finally relayed to the primary visual cortex, also known as V1.
So V1 is like the brain's primary visual processing center.
You got it. But it's important to note that V1 is just the first stop in a complex hierarchy of visual processing areas.
So it's like a team of specialists, each analyzing different aspects of the visual scene.
Exactly. V1 extracts basic features like lines, edges, and orientations. Other areas are responsible for recognizing objects, faces, scenes, and guiding our actions in the visual world.
It's incredible how all these different areas work together to create our visual experience.
It's a remarkable feat of neural orchestration.
Speaking of orchestration, we can't talk about sensory experiences without mentioning hearing. How does the brain decode those sound waves and turn them into auditory experiences?
Hearing, much like vision, involves converting physical energy into neural signals. And it all starts with the ear, which captures sound waves and transforms them into mechanical vibrations.
Right. I remember learning that the real magic happens in the inner ear.
You're exactly right. The inner ear houses the cochlea, a spiral-shaped, fluid-filled structure that's lined with hair cells.
Hair cells. our ability to hear depends on tiny hairs.
It does. These hair cells are incredibly sensitive to vibrations. When sound waves enter the ear, they cause the fluid in the cochlea to move, and that movement bends these tiny
And
hairs.
that bending triggers the neural signals.
Precisely. It opens ion channels in the hair cells, generating electrical signals that are sent to the brain via the auditory nerve.
So it's all about the movement of those tiny hairs.
Ex-
But how does the brain differentiate between different frequencies of sound? How do we know if we're hearing a high-pitched whistle versus a low rumble?
That's thanks to something called the tonotopic organization of the cochlea. Different frequencies cause different parts of the basilar membrane, a structure inside the cochlea, to vibrate most strongly.
So the cochlea is like a frequency analyzer.
Exactly. Hair cells located in different regions of the basilar membrane are tuned to different frequencies.
So it's like a piano keyboard, with each key representing a different frequency.
That's a great analogy. High-frequency sounds activate hair cells at the base of the cochlea, while low-frequency sounds activate hair cells at the apex.
And this frequency map is maintained all the way to the brain.
It is. Different frequencies are processed in different areas of the auditory cortex.
It's amazing how much precision and organization goes into our ability to hear. But hearing is more than just perceiving frequencies. We also need to locate sounds in space.
Absolutely.
How does the brain figure out where a sound is coming from?
That's achieved by analyzing subtle differences in how sound waves reach our two ears.
So our brains are constantly comparing the signals coming from both ears.
Exactly. A sound coming from your right, for example, will reach your right ear slightly sooner and be slightly louder than it is in your left ear.
Those tiny differences in timing and intensity give us clues about the location.
Precisely. And those differences are analyzed by specialized circuits in the brainstem, allowing us to pinpoint a sound's location.
It's like having a built-in sonar system. But, we often take for granted another sensory system that helps us navigate the world,
Which one is that?
the vestibular system.
Ah, yes. Our internal GPS.
It's responsible for our sense of balance and spatial orientation, right?
Exactly. It's what allows us to move through the world without getting dizzy or disoriented.
And this system is located in the inner ear.
Right next to the cochlea.
Wow, so much going on in that tiny space.
It's packed with sensory power. The vestibular system consists of 3 fluid-filled semicircular canals and 2 otolith organs.
And those are also lined with hair cells, like the cochlea.
You got it. But in the vestibular system, these hair cells are stimulated by the movement of fluid within the semicircular canals and otolith organs.
So when we move our head, it sloshes the fluid around, bending the hairs and sending signals to the brain about our movement.
Exactly. The semicircular canals detect rotational movements of the head, like tilting or turning.
And the otolith organs?
They detect linear accelerations, like moving forward or backward, and the head's position relative to gravity.
So the semicircular canals are like tiny gyroscopes, and the otolith organs act like tiny levels.
Perfect analogies.
It's amazing. Where does all that information from the vestibular system go?
It's sent to the brain, where it's integrated with visual and other sensory input to create a stable and coherent sense of our position and movement in space.
It's incredible how all these systems work together seamlessly.
It really is.
Now let's talk about 2 senses that are often linked:
Which ones?
smell and taste.
Ah, yes. The chemical senses.
How does the brain create those olfactory and gustatory experiences?
Oh.
Let's start with smell.
Okay, so smell, or olfaction, relies on olfactory receptor neurons located high up in the nasal cavity in a patch of tissue called the olfactory epithelium.
So when we sniff something, odor molecules are drawn into our nose and bind to these olfactory receptors.
Exactly, and here's the amazing part. There are hundreds of different types of olfactory receptors, each tuned to a specific range of odor molecules.
It's like having a team of scent detectives, each specializing in a different type of smell.
I like that analogy.
So what happens once an odor molecule binds to its corresponding receptor?
It triggers a signal that travels along the olfactory nerve to the olfactory bulb, a structure at the base of the brain.
Okay.
From there, the signal is sent to other brain areas involved in emotion, memory, and conscious perception of smells.
That explains why certain smells can evoke such strong emotional responses and trigger vivid memories.
It's a direct link between our sense of smell and the emotional centers of the brain.
Fascinating. Now what about taste? How does the brain process that sensory information?
Taste, or gustation, relies on taste receptor cells located in taste buds.
And we have taste buds all over our tongue.
Right. Not just the tongue. You'll also find taste buds on the roof of your mouth, in your throat, and even on your epiglottis.
Really? I didn't know that.
Yeah. And these taste receptor cells are what allow us to perceive the 5 basic tastes: sweet, sour, salty, bitter, and umami.
Umami, that's the savory taste, right?
Exactly. And like olfactory receptors, taste receptors are specialized, with different types for detecting each taste quality.
So when we eat something, those tastins, the chemicals that stimulate taste, bind to their matching receptors and send signals to the brain.
You got it. And those signals travel through the brainstem and thalamus before reaching the gustatory cortex.
The gustatory cortex, that's where the brain processes taste.
Precisely.
So it's similar to other sensory pathways, with information relaying through different stations before reaching the cortex for processing.
Exactly. And it's in the gustatory cortex where things get really interesting, because that's where taste signals are combined with information from our sense of smell and other senses to create our perception of flavor.
So flavor is really a multi-sensory experience, combining taste, smell, texture, and even temperature.
It's a symphony of sensory input.
It's amazing how the brain orchestrates all this information to create such a rich and nuanced experience.
It is. And it highlights the brain's incredible ability to integrate information from different sources.
But we've been focusing on sensory input. But the brain isn't just passively receiving information.
Of course not.
It also controls our movements and actions.
That's right. We're talking about motor control, the planning, coordination, and execution of movements.
And this involves a complex hierarchy of structures, right? From the spinal cord all the way up to the cerebral cortex.
Exactly. The spinal cord handles the most basic movements, like simple reflexes.
Like that knee-jerk reflex the doctor tests.
Exactly. Or quickly pulling your hand away from something hot.
So the spinal cord is like a local control center, handling those rapid, instinctive movements.
That's a great way to think about it. And then we have the brainstem, which houses several nuclei that control more complex movements like postural adjustments, eye movements, and facial expressions.
So it's like the brainstem is a step up in the hierarchy, coordinating more sophisticated motor programs.
I like that analogy. And finally, we have the cerebral cortex, the outermost layer of the brain, responsible for the highest level of motor control.
The cortex is like the CEO, making the executive decisions about movement.
Precisely. The cortex handles voluntary movements, planning, and decision-making.
Does the cortex send commands directly to the muscles?
Not directly. The cortex influences movements in 2 ways. It sends signals directly to the spinal cord and brainstem, and it also indirectly modulates the activity of subcortical structures like the basal ganglia and cerebellum.
So it's a complex web of interactions.
It is.
What roles do the basal ganglia and the cerebellum play in this motor control symphony?
Well, the basal ganglia are key for action selection.
Action selection.
Yes, they help us choose which movements to initiate and suppress competing movements that aren't needed at that moment.
So they're like the brain's traffic cops, directing the flow of motor commands.
That's a great way to put it.
And what about the cerebellum? What's its role?
The cerebellum is the master of coordination and fine motor control. It receives input from the cortex, spinal cord, and vestibular system, and uses this information to fine-tune our movements, making sure they're smooth, accurate, and well-timed.
So it's like the brain's quality control department.
Exactly. And the cerebellum is also crucial for motor learning, allowing us to refine our motor skills with practice.
So it's what helps us get better at playing the piano or hitting a tennis ball.
Precisely. It's constantly striving for smoother, more efficient movements.
It's incredible how all these systems work together to produce such fluid and coordinated movements.
It really is.
But think about this. All these incredible motor control systems, all those neural connections, they start as just a simple cluster of cells and develop into this complex machinery.
That's right.
How does that even happen? How does the brain go from a few simple cells to the incredibly intricate organ it is in adulthood?
That's where developmental neuroscience comes in.
Developmental neuroscience.
Yes, this field explores how the nervous system develops from a single fertilized egg into this complex network of billions of neurons.
It's hard to even fathom how such a transformation takes place.
It's a truly remarkable process.
What guides this development? Is it all pre-programmed in our genes?
It's a combination of genetic instructions and environmental influences. Genes provide the blueprint, but interactions with the environment shape the finer details.
So it's nature and nurture working together to shape the brain.
Exactly. And one of the most remarkable things about the brain is its plasticity.
Plasticity.
Yes, it's the brain's ability to change and adapt throughout life in response to experience.
So the brain isn't fixed or static, but constantly evolving.
Exactly. This plasticity is essential for learning, memory, recovering from injury, and adapting to new environments.
So the brain is constantly rewiring itself, optimizing its circuitry based on what we learn and experience. That's incredible.
It is?
Now, speaking of learning and experience, let's dive into the realm of memory. How does the brain encode, store, and retrieve information? There are so many different types of memory. How do they differ?
You're right, there are. A helpful way to think about memory is to categorize it based.
It's a helpful way to think about memory, is to categorize it based on how long information is retained.
Yeah.
So we're talking about those different types of memory: sensory memory, short-term memory, and long-term memory. Could you explain how they differ?
Absolutely. Sensory memory is the most fleeting, just a few seconds. It's like a quick impression of the world, constantly updated as new sensory information comes in.
So it's like a mental snapshot that fades almost instantly.
Exactly. Then we have short-term memory, also known as working memory. This one lasts a bit longer, holding information for maybe a few seconds to a minute. It's like a mental scratch pad where you temporarily store information you need to use right away.
Like when you remember a phone number just long enough to dial it.
Exactly. And finally, we have long-term memory. That's the most durable type, capable of storing information for days, weeks, years, or even a lifetime. This is where we keep all those experiences, knowledge, and skills that shape who we are.
Everything from childhood memories to what we learned in school to how to ride a bike, it's all tucked away in long-term memory. But how does the brain actually convert those short-term memories into long-lasting long-term memories?
That's a question that has fascinated neuroscientists for a long time, and there's no single simple answer. It involves a complex interplay of brain regions and cellular processes.
So it's not as easy as flipping a switch to save a memory for the long haul.
Definitely not. One of the key players in forming new long-term memories is the hippocampus, a seahorse-shaped structure deep inside the temporal lobe.
Ah, the hippocampus. I remember reading about it playing a role in spatial memory, like remembering the layout of a city or a familiar place.
That's right. The hippocampus is crucial for forming new episodic memories. Those are memories of specific events and experiences. And it's not just about storage. The hippocampus also helps us retrieve those memories later when we need them.
So the hippocampus is like the librarian of our brain,
Yeah,
helping us find the right books in the vast library of our memories.
that's a great analogy. But the hippocampus isn't the only player in the memory game. Another important structure is the amygdala, an almond-shaped structure located right next to the hippocampus.
The amygdala, that's the one that's associated with emotions, right?
Yeah.
Especially fear and anxiety.
You got it. The amygdala's job is to attach emotional significance to memories. This makes those memories more vivid and easier to recall. For example, a frightening experience is more likely to be remembered because the amygdala attaches a strong emotional tag to it.
So the amygdala is like highlighting or bookmarking those important memories,
Mhm.
making sure we don't forget them.
Exactly. But this brings up another fascinating question. How does the brain physically store memories at the cellular level?
Right. How can something as abstract as a memory be encoded in the physical structure of the brain?
That's where things get really mind-blowing. I think you were on the right track earlier when you mentioned synaptic plasticity, the idea that connections between neurons can strengthen or weaken based on experience.
So the more we use a particular neural pathway, the stronger that pathway becomes.
Exactly. One of the best studied examples of this is long-term potentiation, or LTP.
Ah, LTP. It's a long-lasting increase in the strength of a synapse, right?
Precisely. LTP was first discovered in the hippocampus, but we now know it happens in other brain regions as well. We believe it's a crucial cellular mechanism for forming new memories.
So when we learn something new, LTP is happening in our brains, strengthening the connections between the neurons involved in that new information.
Exactly. And it's not just about strengthening connections. Synaptic plasticity also involves weakening those connections that are no longer needed. It's like pruning a garden, getting rid of the unnecessary branches to make way for new growth.
So the brain is constantly remodeling itself, optimizing its circuitry based on what we learn and experience. That's incredible. but I imagine LTP is just one piece of the memory puzzle.
You're absolutely right. There are many other molecular and cellular processes involved in how our brains encode, store, and retrieve memories. It's a complex and fascinating area of
It
research.
really is. But we can't talk about the brain without acknowledging that it faces challenges throughout life. What about aging and those neurodegenerative diseases we hear so much about?
Aging is an inevitable part of life. And just like other parts of our body, the brain undergoes changes as we age. Some of those changes are normal, while others can lead to cognitive decline and increase the risk of neurodegenerative diseases.
So even the mighty brain isn't invincible.
Unfortunately not. But the good news is that there are things we can do to help protect our brains as we get older. A healthy lifestyle, regular exercise, a balanced diet, and engaging in mentally stimulating activities, those can all go a long way in keeping our brains sharp.
So it's like taking care of our brain is just as important as taking care of our physical health.
Absolutely. And one of the most devastating neurodegenerative diseases is Alzheimer's disease, which gradually steals people's memories and cognitive abilities.
Alzheimer's is heartbreaking, affecting so many people. What do we know about what causes it, and if there are any potential treatments on the horizon?
Alzheimer's is a complex disease, and scientists are still working to unlock its mysteries. We know it's characterized by the buildup of abnormal protein deposits in the brain, primarily amyloid plaques and tau tangles.
I've heard those terms before, amyloid plaques and tau tangles. How do they actually contribute to Alzheimer's?
Well, amyloid plaques are clumps of a protein called beta amyloid that accumulate between nerve cells. These plaques disrupt communication between neurons. Tau tangles, on the other hand, are twisted fibers of a protein called tau that form inside neurons. These tangles interfere with the normal functioning of the neurons.
So it's like these protein deposits are gumming up the works of the brain, preventing those critical communication pathways from working properly.
Exactly. And over time, this damage leads to the death of neurons, resulting in the progressive cognitive decline we see in individuals with Alzheimer's disease.
It's a tragic chain of events. Are there any treatments that can slow down or reverse this process?
Unfortunately, there is no cure for Alzheimer's yet, but there are treatments that can help manage the symptoms and potentially slow down the progression. Researchers are hard at work trying to develop new treatments that target the underlying causes of the disease, such as preventing the formation of amyloid plaques or tau tangles. It's a challenging area of research, but there's hope.
It's inspiring to know that scientists are dedicating themselves to finding solutions. Shifting gears a bit, I'm curious about the brain's regenerative capacity. Can the brain actually repair itself after an injury or disease?
That's a great question. The brain's ability to regenerate is a topic of ongoing research and discussion. We know the adult brain has a limited capacity for generating new neurons, a process called neurogenesis.
So the brain isn't as good at regenerating as other organs, like our skin or liver.
That's true. But here's the exciting part. We're learning that the brain is much more plastic than we initially thought. Even in adulthood, it can rewire itself and adapt to new challenges, even after an injury.
So there's hope for recovery after something like a brain injury or stroke.
Absolutely. Rehabilitation therapies, such as physical therapy, occupational therapy, and speech therapy, can help stimulate the brain's plasticity and promote recovery.
It's encouraging to know that the brain has this inherent ability to heal and adapt. As we wrap up our deep dive into neuroscience, I'd love to touch on one of the most profound and mysterious aspects of the brain, consciousness.
Ah, consciousness. The subjective experience of being aware of ourselves and the world around us. It's a concept that has puzzled philosophers and scientists for centuries.
And I imagine we're still a long way from fully understanding how consciousness arises from the physical matter of the brain.
We are. However, neuroscience is making progress in identifying the neural correlates of consciousness. In other words, we're beginning to pinpoint the brain activity that's associated with different states of awareness.
So we're starting to map out the regions of the brain that are involved in generating our conscious experience.
Exactly. For example, we know the prefrontal cortex, a region involved in planning, decision-making, and self-awareness, is highly active when we engage in conscious thought.
So the prefrontal cortex is like the conductor, the consciousness orchestra.
That's an interesting way to think about it. But it's important to remember that consciousness is a complex phenomenon. It's not localized to a single brain region. It likely involves a complex interplay of many areas working together in ways we're still deciphering.
It sounds like consciousness is the ultimate brain teaser. The more we learn about the brain, the more we realize how much we still don't know.
That's what makes neuroscience so exciting. It's a field that's constantly evolving and new discoveries are being made all the time. The more we learn, the more we appreciate the incredible complexity and wonder of the human brain.
That's such a great point. And speaking of incredible discoveries, the book's discussion of those early experiments on squid axons really stood out to me. It's amazing how something as fundamental as the action potential was a mystery for so long.
It is fascinating, isn't it? Those pioneering studies paved the way for the incredible advancements in neuroscience we're seeing today.
It's a testament to the power of basic research, pursuing knowledge simply for the sake of understanding. We never know which seemingly small discoveries might lead to major breakthroughs in our understanding of the brain and how to treat neurological disorders.
I completely agree. Those early researchers working with squid axons likely had no idea the profound impact their work would have on the field.
Speaking of impact, this deep dive has definitely left me feeling a bit overwhelmed. The brain is such a complex and powerful organ, it's humbling to think about.
It is humbling, isn't it? And it reminds us that we're still just scratching the surface when it comes to understanding how this incredible organ works.
It's true. But even with all the mysteries that remain, I think it's safe to say that the brain is the most fascinating and enigmatic frontier in all of science.
I couldn't agree more.
It really is incredible how much is going on up there that we have no clue about. This deep dive honestly, it sparks so many more questions for me.
That's the beauty of neuroscience, isn't it? The more we learn, the more we realize how much more there is to discover.
It's true. For example, the book talks about brain lateralization. That's where certain functions are predominantly controlled by one hemisphere of the brain, right?
Ah, yes. Lateralization. It's a fascinating concept. We always hear about people being left-brained or right-brained.
Yeah, like the whole left brain people are more analytical and logical, and right brain people are more creative and intuitive. How much truth is there to that?
While there's some evidence that certain functions tend to be lateralized, like you said, the idea that people are dominated by one hemisphere or the other is a bit oversimplified. The two hemispheres are constantly communicating and working together, even for tasks that we might think of as being more associated with one side.
So it's not as black and white as we often hear.
Exactly. It's much more nuanced than that. Take language, for example.
Uh,
We know that language processing is mainly handled by the left hemisphere for most people, but the right hemisphere also plays a role in understanding things like the emotional tone of language and interpreting those nonverbal cues?
that makes sense. After all, communication is more than just words.
Exactly. It's about tone, facial expressions, body language, all those things.
Right, right.
And that's why both hemispheres contribute to our overall understanding of what someone's really saying. Lateralization is a fascinating area of research because it really highlights that specialization, but also the integration of different functions within the brain.
It's like different departments in a company, each with their own specialties, but they have to work together to achieve a common goal.
A perfect analogy.
But shifting gears a bit, I'm curious about the connection between neuroscience and artificial intelligence. It seems like a hot topic these days.
It is. As we understand more about how our brains work and their computational abilities, well, it's definitely influencing how we're developing AI.
So we're basically using what we've learned about the brain to try to build more intelligent machines.
Exactly. For example, the concept of neural networks, which is a type of machine learning algorithm that was inspired by the structure and function of the brain, and it's revolutionized AI.
That's incredible. So the brain is serving as a model for creating more sophisticated AI.
It is, and the flow of information goes both ways. As AI systems become more complex, they're actually giving us new tools and insights that we can use to study the brain itself.
It's like a feedback loop,
It is.
where each field is pushing the boundaries of the other. But I'm wondering, as we learn more about the brain, does it change how we think about things like, I don't know, free will and responsibility? Because it's kind of a big question.
That's a huge question, and one that both neuroscientists and philosophers are still grappling with. As we uncover more about the neural processes underlying our thoughts, emotions, and actions, it does raise some really interesting questions about the nature of free will.
Right. So, like, if our decisions are ultimately just a product of all this neural activity, does that mean we don't actually have free will?
Well, it's not necessarily that simple.
Okay.
Some argue that we might need to redefine what we mean by free will, given what we're learning. Others argue that just because our decisions have a biological basis doesn't mean that they're not freely chosen. It's a very complex and nuanced debate.
It really is. It highlights how neuroscience raises some truly mind-bending questions about what it means to be human. Well, this has been an absolutely incredible and thought-provoking deep dive into the world of neuroscience. We've covered so much: the fundamental building blocks of the brain, those intricate systems that govern our senses and our movements, the complexities of memory and consciousness, and even, touched on some of the philosophical questions that neuroscience raises.
I agree. We've barely scratched the surface, but hopefully this has sparked a sense of wonder and curiosity in our listeners.
Exactly. There is so much more to explore, and the field is constantly evolving. So until next time, keep those brains engaged and never stop asking questions.
I second that.
Thanks for joining us on this deep dive into neuroscience. We'll see you next time for another fascinating exploration.